So welcome back to our next video here. Last video, we covered the structure of DNA. Today, we're going to talk about DNA replication. So replication essentially means that you're making another copy of the DNA. As we mentioned in the last video, all of us started with just our one kind of single copy of DNA. And obviously now you have a lot more cells, so you need a lot more copies of DNA. So replication is how we go from just one single copy to all of these copies in all of your cells. And so what happens is we think about the structure of DNA. We have all of these little nucleotides. We have our A's and G's and C's and T's. And it's very, it's great. It looks like this nice ladder. And last time we talked about how we can easily unzip the middle of the ladder. And we can expose now all of these bases. And so the first step in replicating DNA is to unzip the DNA, just like that.
Now, once that's unzipped, and you can see here on my cardigan and on Chemeter's model, these teeth, or the rungs, the two sides of the rungs over there in Chemeter's model, are exposed. Exposed. They don't like that, okay? They want to pair up with something, but as we already know, A can only really ever pair up with T, and G can only ever really pair up with C, physically, like puzzle pieces that don't match. So they will start, there's free-floating nucleotides that we made earlier, okay, before the cell went to replicate. And those free-floating nucleotides will come in and they will match up. It'll be helped by enzymes, kind of like Chemeter's hands and enzyme, right? It's going to help that process out. But they're going to match up one after the other on both sides of the zippers here, which we'll see in a little bit. Now, on to Chemeter.
Yeah, well, and this last part too, there's more to it. When you get into this in depth, if you take biotechnology, I'm just going to do a quick plug for biotech. We get into DNA structure and replication in another layer of detail, which is really cool. And anyway, when all of this is said and done, that really solid, strongly bound together backbone, those bonds need to be finished up. So that's what enzymes come along and do. They make sure that that backbone, the sugar phosphate backbone, is all bound together, and then you've got your new strands, two new strands of DNA.
So here is a slow motion kind of animation of what's happening. So if you can imagine on the bottom here, we've unzipped our DNA. We have kind of two halves of our DNA. Imagine we're only looking at kind of one on the bottom here. And as these things reset, we'll give it a second here maybe. There we go. You can see the nucleotides kind of floating on in, pairing up with their kind of appropriate base. So we can see a G matching up with a C there. We see our phosphate bond or phosphate sugar backbone kind of being bound together as well. And so this process keeps happening. So now we have a T joining with an A, and it'll continue to fill in all of these gaps. And if you imagine now we have this going on our other half of DNA, you can see how we would result with two new kind of ladders, two new strands of DNA.
And I'll just share another representation of this. So we've got our model of DNA over here on the board. Sugar phosphate backbone, rungs in the middle. These are our bases, where the letters are being represented right on them, right, and they're color coded. But anyway, step number one, we said the hydrogen bonds down the middle of the double helix break, and they're opened up. So enzymes do this. We're opening up our strand. As the strand opens, now we have exposed bases down here. Enzymes are going to help guide these nucleotides into place. You also have to imagine that this is taking place at the molecular level. Not if you're you. This is taking place at the molecular level, and this is driven in part by random motion of the molecules. So these are all moving around. Just imagine that. And eventually, we get G is going to come in here. Again, there's an enzyme that's helping guide this, but there's a G that matches with this C. And then we've got the same thing happening on the other side here. We've got this G is pairing with the C there. And there we've got a T is going to match up with that. And then we've got another C going in there. I got these U's in here. Get out of here, U's. That's going to match up there. It's going to continue on the other side. This is where one of those details in DNA is that we're not going into here. There's a direction that this happens in, so I'm not obeying that rule right now. So if you know more, Please forgive me for not following the 5' 3' directive.
I knew he was going to name drop that.
All right, so now we've got the start of two models. We're just going to continue splitting them apart here. And if you've got the time, you can watch me struggle to put the rest of this together here. There's going to be a G there, and now I need another C. Oops. See, it won't happen. It doesn't happen, so that one has to go away. Now I need a G.
Now, can you show us the end of those things? That they've actually got shapes on those models, don't they?
Yes, they do. Look at this.
So that's why it wouldn't actually fit.
C, and then here's a C, and oops, there we go. Those two fit together. If I have an A and a C, they don't fit. It's kind of like what's that cube thing that little babies have?
The where you put the pegs in the holes,
Yeah,
a round peg in a square hole.
Mm-hmm. It's kind of like that. All right. And I'm sorry I'm taking the time to go all the way through here, but what's nice is at the very end, we will see that we've got two identical strands.
Just have to add some suspenseful music during this.
Yeah.
I wonder if I can speed it up and put it to like Benny Hill music or something.
That would also be fun.
I would appreciate that. There we go. Look at this. T A G C C G G C T A T G C A T C G. Identical. Oh my gosh. That's how your cells get their copy of DNA from the original cell. It's also how you make copies of your DNA if you're going to be passing it on to your kids someday. Yeah, make identical copies, and all it does is base pairs is the backbone of all of this. A's with T's, G's with C's. Grumpy cat.
And so here's this model here shows the same thing. So you got it in your notes. Do you guys want to watch that animation of how fast this happens in real time?
Yes.
Yes.
So you don't have to mess around with how slow it takes Chemeter to do it.
Right.
No.
That was too slow.
So here's, again, there's more detail to all of this, but down in the lower left, now it's coming in from the left side, that is the original strand of DNA, and it is being unwound by this blue enzyme there. So as it goes in, it's unzipped. That's like the tab of the zipper, unzipping the two strands. One strand's going off to the right, the other one's going off to the left. And as they're going off to the sides, these other proteins, enzymes there, they are grabbing free nucleotides that are just in the environment. They're floating around nearby. And if they match, they get added to the strand. If they don't match, they're chucked out, and then another one is tried. Now, as far as the speed of this goes, what is the enzyme that's unzipping the DNA is spinning around it's the speed of a jet engine, is it not, Mr. Hamele? That's what they...
I remember that, but I don't want to be quoted on it.
I think it's, I'm going to quote it. It's spinning at the speed of a jet engine. Just think about that, how fast that is moving, and your DNA is being replicated at like that speed, the speed of a jet engine. Your DNA is really long. We got 3 billion base pairs to get through, but still, that's pretty darn fast.
Well, is that it?
I think that's it. Basics, but that's really all we need.
Sounds good.
All right.
We now know the structure of DNA, and we know how DNA is able to take advantage of some of the quirks of that structure, the strong backbone and the weak middle bonds, and the fact that only A's and T's and G's and C's.
And that's
That's it.
that's it for Mr. Hamele.
Oh no.
I'm sure he was going to say something lovely, but A's and T's, G's and C's, base pairing is at the core of this. The sequence of the bases is going to be the information, and then replication. That's how we can make the DNA for the rest of our cells as we grow, or this is how organisms pass it down from generation to generation. For Mr. Hamele, myself, and Miss Ripley, thank you very much for watching.
We'll see you next time.
Can I let me try to annotate here real quick. Can I?
Oh no, he's gone.
Anyway, all right. That should be a good enough ending.